So today we're going to talk about techniques you'll need for the plant module. The first and most important thing is to label your plates that you're given. So you're going to want to divide your plate into quadrants, the first two being a little bit larger than the second two because you'll have more infected plants than you will uninfected plants. Now each of these quadrants is going to contain a different plant or plant infected with bacteria, so you'll want to make sure you label appropriately either your Columbus strain or your Landsberg strain with your two different bacterial strains. So once you label appropriately, you'll be ready to start isolating your plants. So here is our magnified view of a seedling. To begin at the base, we have the root, which then meets the hypocotyl here, which is the stem of the plant. Between the root and the hypocotyl, we see that there is the seed coat, where the seedling emerged from. This can be located at this connection here, but I've also seen it up at the leaves and other areas. The hypocotyl will connect to the shoot area, which is where the leaves come off of. These two specific leaves are called the cotyledon. They are the first two leaves that emerge from the plant. The remaining leaves and the stem for the flowers will emerge from the shoot here. When you go to cut your plants, you want to cut right at the base of the hypocotyl here to remove all of the roots. So you want to cut in this area. It's okay if you accidentally get a little bit higher, but you don't want to cut any part of this top here, or the shoot will, or the seedling will no longer survive. When you pick it up, you're going to want to use your tweezers to isolate the leaf or the cotyledon when you pick it up. It does not work to pick it up by the tiny stem or the hypocotyl. It's important to remember sterile technique. Even with these plants, bacteria and other fungus can grow on these plates as well. So you always want to keep your plates closed unless you're working with them. You'll be given autoclaved, sterilized tweezers as well as a sterile scalpel. When you're not using your scalpel, put it into the sterile 50 ml conical that we have at your bench. So the first thing you want to do is check out your plants and find ones that are isolated and easy to remove and cut. So what you'll want to do is with your tweezers, make sure you find a plant that you feel is isolated and ready to go. Sometimes it helps to kind of lift it up a little bit so that the plant is loose from the agar. You need to be gentle when you do this. If it's hard to see, you can use the dissecting microscope. Once you're ready to cut your plant, you're going to cut it between the hypocotyl and the root base. Basically right at the base of the stem. It's probably best not to talk while you're doing this because again you can transfer germs onto your plate. You'll want to make a small cut and be careful not to cut the other plants in the surrounding area because you'll need those. Alright. It is not as easy as it seems. Once it's cut, make sure to cover your plants. You're going to transfer this seedling that has been cut. You want to do all your controls first. So this is my uninfected control. And when you transfer, you need to make sure that the cut piece is touching the agar. Alright, now let's go on to our uninfected control. Our infected plant. We will again need to isolate a plant with our tweezers. Gonna lift it away a little bit. Cut off. Return our scalpel. Now I have a cut plant that I need to dip in our bacteria. So for this, it's okay whether it's an isolated colony or a continuous streak. You just want to graze the colony with the cut end and place this onto your plate. Again, you want to make sure that the cut end is touching your agar. Once that, you can continue cutting your plants. Make sure you put them in the right places so that you know you have infected with the bacteria that you have labeled.